Hello everyone. So today is our first class on this course. So, I don't see anyone has joined. So, I will record this session and I will upload the session in that NPTEL site. So, as this is first class, so first I want to introduce myself, and if some of you is there, then I would like to get an introduction from you side also. So today also we will discuss about the course, like what is this course and what is it all about, and we will discuss like when is the exam of this course and how it will be going. So before this, I will like to wait for some 5 minutes. So if somebody is there, then he or she can join. For like we will keep this call on hold so that we can wait for the other students to join. So okay, let's wait for some time. So we will start the class at 6:25 after some 4 minutes. So So this is 6:25. So now we will start the class. I can't see somebody join, so I will record this section. I will record this session and I will upload on the NPTEL site. So, okay. Hi everyone. So myself Shivangi. I am from Indian Institute of Science, Bangalore. So I'm a chemical biologist. So my work is basically on the synthesis of anti-cancer agents. So I am also working on the interface of chemistry and biology. So this course is also it's also not purely biology, and it's also not about pure chemistry. So this course is also at the interface of chemistry and biology. So yeah, this is an interdisciplinary course. So in this course, we will study about the biomolecules. So biomolecules is basically uh, these are the molecules which are present in your body. These are the, like we can say, organic molecules, or the molecules which will carry out the chemical transformation in your body. We will call it as biomolecules. Biomolecules like peptides, these proteins, vitamins, carbohydrates, fatty acids, DNA, RNA, all these are the biomolecules, and these biomolecules are present in your body, and these biomolecules are very important and is sorry, and these biomolecules are very important as it is required to carry out the various important chemical transformations in the body. So I would like to share my screen in which we will discuss about this course. So I think you are able to see my screen. Yeah, so now we will see about this course. So this course is all about the so this course the full name of this course is Essential of Biomolecules, Nucleic Acid and Peptides. So this nucleic acid and peptides are the biomolecules, and this course is why these biomolecules are essential and how they are carrying out the chemical transformation in human body. So the proposed course aims to provide essentials of chemistry and biology of two very important class of biomolecules, nucleic acid and proteins. So this course will be at the interface of chemistry and biology, and it will tell about the two important class of biomolecule. One is nucleic acid, which involve DNA and RNA, and the proteins. Um, for these are the important two topics of this course. So this course allows to decipher how structural features are translated into biological function, how highly organized and selective chemical reactions are adopted that allow DNA to replicate or dictate stepwise synthesis of specific sequence of proteins. So in this they are telling like in what all we are about to read in this topic. So first is how structural features are translated into biological functions. Second is how highly organized and selective chemical reactions are adopted that allows DNA to replicate to dictate stepwise synthesis of specific sequence of proteins. Third is how organic chemistry tools in combination with enzyme were ingeniously applied to determine sequences of DNA and proteins, and how chemical modification could be done to mimic similar biological properties. This course also includes modern techniques, development of biomolecular probes as high throughput detection of biomolecules, single nucleotide polymorphism, and disease diagnosis. Overall, this course falls within the domain of organic chemistry and biology, as I told you. So, this course is basically taught by Professor Lalmohan Kundu from IIT Guwahati, and these all are the pattern like how we are going to read in this course. This is week 1, week 2, week 3. In week 1, we will read about introduction, nucleic acid and proteins. Second is synthesis of nucleobases, nucleotides and oligonucleotides. Week 3rd is solid phase synthesis of oligonucleotides. Week 4 is DNA replication, polymerases, DNA sequences and PCR. Week 5 DNA damage, mutations and cancer. Then week 6 is related to DNA to proteins, transcription, translation and genetic code. And week 7 is all about peptides, sequencing and application in therapeutics. Week 8 is solution phase and solid phase peptide synthesis. Week 9 is expansion of genetic code, DNA, RNA and molecular probes. Week 10 is modern techniques for biomolecules and disease diagnosis. Week 11 is structure and chemistry of sugars and carbohydrates. And week 12 is carbohydrate based polymers as biomolecules, probes, and therapeutics. And in last week, he will conclude his all the talks. And the books and references he used to give his lecture, first is Essentials of Chemical Biology by A. Miller and J. Tanner. The second is Biochemistry by Berg, Timko and Stryer, and these all are the books and references which you can use, which you can use to read. And if we will see the other things, 1 minute. I think so. Huh. So this course is 12-week course, and the course starting date is 22nd January 2024. So this is basically first week of your course. Yesterday it has been started, and our TA class will be on each like every Tuesday from 6 to 8 p.m. The end date of this course is at 12th April 2024. This course will be end, and exam will be on 27th April 2024. So this is from January to April. Exam will be on 27th April, and yeah, so exam will be on 1 minute. Huh. So, yeah. So the course, like, uh, as you all know, that professor will be teaching this course every week, like every day of the week. But I will be taking the PA class every Tuesday from 6 to 8 p.m. If any of you are feeling some doubt regarding the course, regarding some topic, or in any question, then you can come to me. We will discuss that doubt in the doubt session class, and I will try to solve the assignments and the MCQ question as much as possible. So we will discuss about the assignments in this class and your doubts. So as this is an interactive class in which you people can ask the doubts, so I will request you to all to join this TA class so that we can interact and discuss many things regarding this course. So the what else is, I think for regarding this course, it is sufficient. I introduced myself also. So now I will like to discuss about the assignment. Assignment. So 1 minute, I will share the assignment with you people. Yeah, so this is week first assignment. We will discuss about this assignment. I will request you all. So basically, he is giving lecture. This course is very good, and it is at the interface of chemistry and biology. No one will feel if we are having interest in chemistry also, then also this course will be interesting. And if somebody has interest in biology, then also this course is interesting. And also, it will tell about the fundamentals of biology. So this course will be helpful for all of you, and also it will be helpful in your net gate exams. So I will request you all to carefully attend his lecture so that we can discuss the doubts and questions in this lecture. So I will request to you to listen his lectures so that you will be able to solve the assignments properly in this lecture. So this is big first assignment which I am going to discuss today. So let's discuss the first week assignment, essential of biomolecule, nucleic acid and peptides. So going to the first question. So the first question is, among the following, the biomolecules are. So as I told you, so what are the biomolecules? One minute. Hmm. So among the following, the biomolecules are. So as I told you, the biomolecules are the molecules which are present in the human body and are necessary to carry out some of the chemical transformations in the human body. So tell me which are the biomolecules among all. Option A is DNA, option B is HCl, option C is cyclohexane, and option D is vitamins. So which is the right option? As you can see, HCl is an inorganic acid, cyclohexane is an organic molecule, whereas DNA and vitamins are the biomolecules present in the human body. Yeah, so A and D. So A and D, DNA and vitamins are the biomolecules present in the human body. So this is the right answer. So moving to the next question is, can the nucleobases be synthesized in the laboratory? So nucleobases are basically, what are the nucleobases? So if we will see, nucleobases are the bases which present in the DNA. So DNA is basically composed of nucleobases, sugar and phosphate groups. You see, DNA is composed of nucleobases, sugar and phosphate group. And nucleobases are further of two types, purines and pyrimidines. So if you will see, I will show you nucleobases. Yeah, so these are the nucleobases. Nucleobases are the nitrogen containing compounds that form the basic building block of the nucleic acid. So these are the uh, nucleobases. So these two, we are having two rings, and these are the purines. And whatever it is having three bases, which is having one ring. These are the pyrimidines. So these are the nucleobases which leads to the formation of DNA, and these if you will see the structure, we will see the structure, then we can analyze that these nucleobases can be synthesized in the laboratory. So yes, these are established. Yes, there are established procedure to synthesize the biomolecules in the laboratory. So yes, nucleobases can be synthesized in the laboratory. So the among the following, the constituent of RNA is. So DNA is full form of DNA is deoxyribonucleic acid, and RNA is RNA is uh, ox, DNA is deoxyribo, RNA is oxyribose nucleic acid. So in this deoxy is not present, so this is not present. Also adenine, thymine, cytosine, and guanine. These all are the four nucleobases which are present in the DNA. Wherever in the case of RNA, thymine is replaced with that of the uracil. So in RNA, adenine is present, uracil is present, guanine is present, and cytosine is present. So among all, thymine is present in the RNA, uracil is present, and guanine is present. So B and D is the right answer. So uracil and guanine are the constituents of RNA. However, guanine is present in DNA also, but thymine and deoxyribose are present in DNA only. So these all are the right answers. Okay, so uracil and guanine, these two are the constituents of RNA. However, guanine is present in DNA also, but thymine and deoxyribose, these are the constituents of DNA. So B and D are the only constituents which are present in the RNA. So the fourth question is peptides are primarily composed of. The first option is A, B is nucleobases, C is amino acid, and D is lipids. So peptides are basically the, we can say that peptides are proteins. So peptides are composed of amino acids. So amino acid, the polymerization of amino acids lead to the formation of peptides. So C is the right option. Peptides are primarily composed of the amino acids. So moving to the next question, nucleobases are found in. I already told you nucleic acid, DNA and RNA are composed of nucleobases. So nucleobases are found in DNA and RNA. Yeah, so moving to the next question. So the next question is the anomeric carbon in glucose is. So in this, the anomeric carbon is the carbon which is attached to the two oxygen atom. If you will see, carbon 2 is attached to one oxygen, 3 is 1, 4 is one oxygen atom. Here that is also attached to one oxygen, whereas carbon 1 is attached to the two oxygen atom. So carbon 1 is the anomeric carbon. So C1 is the anomeric carbon in the glucose. So the complementary strand of the following uh, uh, the complementary strand for the following sequences, we have to tell the complementary. So as we all know, A is adenine, T is thymine, G is guanine, C is cytosine. So A will bind to the thymine, whereas G is bind to the cytosine. So A, here it will come as T, again A, C, G, A, G, T, C, A, T. So the option is T, A, C, T, A, C, T, A, C, G, A, G, T, C, A, T. So option A is the right answer. Okay. So option A is the right answer. A is going to pair with T, thymine, whereas G is going to pair with C. So in a double-stranded DNA, the number of moles of thymine is 0.25, whereas the number of moles of guanine is 0.30. So the number of moles of cytosine and number of moles of adenine respectively. So we should know, in a double-stranded DNA, moles of number of moles of A is equal to number of moles of T, whereas number of moles of G is equal to number of moles of C. So it is asking us thymine is 0.25. So thymine is 0.25. So adenine should also be 0.25. And guanine is 0.30. So the cytosine should be 0.30. So the answer is 0.30 and 0.25. So option C is the right option. 0.30 and 0.25. You can note down. So the next question is the weight of cytosine and adenine molecules are. In the question 2, they have given the number of moles. Number of moles for cytosine is 0.30 and for adenine it's 0.25. That is N. So here they have given molecular weight and we have to tell the weight. So number of moles is equal to given weight upon molecular weight. So you have to calculate this. So you can calculate as number of moles multiplied by molecular weight. So weight of cytosine is equal to number of moles of cytosine is 0.30, molecular weight of cytosine is 111. So triple one multiplied by 0.30, 33.3. So there is no need to calculate even second one because cytosine if we will calculate only this is having 33.3. Option A is the correct option. Same you can calculate for adenine also. The next question is ATP is the source of energy in living cells. The nucleobase present in it is. So if you don't know the even the structure of this nucleobases, you should if you will remember that adenosine trinucle adenosine tri adenosine triphosphate. So adenosine is the nucleobase present in the ATP. So adenine is the nucleobase present in the ATP. So option D is the right option. You can see. So among the following which will have higher melting temperature. So a double stranded RNA, double stranded DNA, or double strand between DNA and RNA. So we all know double stranded DNA is having the high melting point. So I'm expecting you know that what is the melting temperature. So the melting temperature is the temperature at which 50% of DNA is converted into single-stranded DNA. 50% of double-stranded DNA is converted into single-stranded DNA, or we can say that 50% of double-stranded DNA will be denatured. That is called the melting temperature of DNA. So the number of major grooves in a double-stranded DNA of 1024 base pair is, so if you will see the number of base pair present in one major groove is 10 base pair. So if you will see 1024 base pair and number of base pair present in the major groove is 10, so we can say that 102.4 are the number of major grooves. Yeah. So this is the right answer. So at pH 10 the thymine will exist as, so this you should know. So thymine then, um, yeah. So thymine, the pK of thymine is 10.47. 10.47. That is the pKa. pKa, and the first physiological pH of body is 7.4, which means 7.4 is acidic as compared to 10.4. So thymine is present in the acidic medium now, so it means it is protonated. So both the NH will be protonated. So option A is the right option. So thymine will exist as this protonated. So option A is the right answer. So these all were the questions, but you have to see. If you are feeling any doubt, then you can come to me. We will solve this question. So this is all about the assignment. I think these were the two assignments. So I will upload this assignment also, and I will upload this video also in the NPTEL site, and also I will make a folder in which I will upload all the weekly assignments. So you can go through these assignments, and if you are feeling any doubt, then you can come to me. We can solve the doubt. Okay, so now we will meet in the next class. So the next class will also be on Tuesday from 6 to 8 p.m. same timing. So okay, for now, uh, this is done. So we will meet in the next class next Tuesday 6 to 8 p.m. So okay, I'm leaving this class now. We will meet in the next class.